This is the second part of chapter respiration in plants. So in the previous part, glycolysis was discussed and anaerobic respiration, fermentation, lactic acid fermentation, alcoholic fermentation was discussed earlier. So in this part 2, we will discuss about aerobic respiration. Pyruvic acid enter into mitochondria, and here pyruvic acid is completely oxidized to production of it will lead to the production of carbon dioxide. Pyruvic acid will convert it into carbon dioxide plus energy. So first process to for the first process, complete oxidation of pyruvic acid by the stepwise removal of all hydrogen atom leaving 3 molecule of CO2 take place in matrix of mitochondria. Second, the electrons are passed from you can say hydrogen atom to molecular oxygen with the simultaneously synthesis of ATP in the inner mitochondrial membrane. So pyruvate further first enter into the mitochondrial matrix, and here pyruvic acid is converted into acetyl coenzyme A first with the help of an enzyme named as pyruvate dehydrogenase. And you can see this is the tricarboxylic acid cycle, TCA cycle, and Krebs cycle. Another name of this cycle is Krebs cycle. You can see in this cycle is from NCERT. Pyruvate is converted into first acetyl coenzyme A. This acetyl coenzyme A will enter inside the Krebs cycle, and it will combine with oxaloacetic acid to form citric acid. Citric acid is converted into alpha ketoglutaric acid. Alpha ketoglutaric acid is converted into succinic acid, and succinic acid further converted into malic acid, and malic acid at the end will again form oxaloacetic acid. So these are the cyclic events or cyclic step that take place, and in this process there is production of NADH, and NADH will take place in alpha ketoglutaric acid step, and in succinic acid ATP and NADH will form, and in malic acid area FADH will form, and in oxaloacetic area NADH again form. So these are the major steps in which we can see that NADH and ATP formation and FADH formation also take place. So let's start with these points. First, condensation of acetyl group with oxaloacetic acid and water will take place with the help of citrate synthetase enzyme, and citric acid will form, and citrate will further converted into isocitrate. Here decarboxylation will take place. Decarboxylation means carbon dioxide is removed, and it will lead to the formation of alpha ketoglutaric acid, and alpha ketoglutaric acid further converted into succinyl coenzyme A. Succinyl coenzyme A further converted into succinic acid, and succinic acid will further converted into you can say with the help of energy currency will further enter into Krebs cycle again. So during Krebs cycle there is a production of 2 molecule of CO2, 3 NADH, 1 FADH and 1 GTP, and this Krebs cycle will take place in mitochondrial matrix area. So during the whole process of oxidation of glucose, 10 NADH, 2 FADH and 2 GTP or 2 ATP will form. Next is electron transport system or oxidative phosphorylation, you can say. So this is the metabolic pathway in which electrons are transferred from one carrier to next carrier, and this electron transport system is present in mitochondrial membrane, inner mitochondrial membrane. Electron transport system comprises the following complexes: complex number 1, 2, 3, 4. Complex 1 contain NADH dehydrogenase. Complex 2 contain an enzyme known as succinate dehydrogenase. Complex number 3 also contain a complex known as cytochrome BC complex, and complex number 4 is cytochrome A1 and A3 complex. Complex number 5 is named as ATP synthase complex. You can see in the corner area, this is the electron transport system in diagrammatic view. This diagram is from NCERT. You can see the first is intermembrane space, then you can see inner mitochondrial membrane and the matrix area. In inner mitochondrial membrane, these complexes means complex number 1, 2, 3, 4, 5 are arranged in a chain like sequence attach with one complex to second, then third, then fourth, and then fifth. And you can see from FMN, the hydrogen or the proton are pumped from matrix to intermembrane space. And here in ubiquinone, protons are also pumped from matrix to intermembrane space, and two electrons are transferred. You can see FMN transfer the two electron to FES, FES will transfer the two electron to Q, and then Q will transfer the electron towards cytochrome B, ferrous sulfate, Q, cytochrome C, and cytochrome A and A3. So here we can see that electron is passed from one carrier to the next carrier present in complex 1, 2, 3, 4, 5, and protons are also pumped from matrix to intermembrane space with the help of different enzymes. Next, the ubiquinone is then oxidized via cytochrome C. The gain of electron and loss of electron is known as oxidation and reduction. So in this you can see the complex 1, 2, 3, 4 are first gain of electron will take place, then the electron is transferred to next complex, then loss of electron will take place. So these complexes will oxidize, reduce, oxidize, reduce, oxidize, reduce in a cyclic manner, and electron is transferred from one complex to the next complex, and the proton is transported. So the cycle is explained in theoretical portion in the first half area, you can see. So it is very important to note down that one NADH molecule will give rise to 3 ATP, and one molecule of FADH will give rise to 2 ATP. It is very important and very crucial step. We must remember that one NADH is equal to how many ATP? 3 ATP, and one FADH is equal to how many ATP? 2 ATP is there. And oxygen play a vital role in removing electron. Next, this is the fifth complex named as ATP synthase complex. ATP synthase complex contain two major portions, F naught and F1 particle, and in F naught area protons enter through F naught and exit from F1 particle into the matrix area. So these ATP synthase complex is present in inner mitochondrial membrane. So energy released utilized in synthesizing ATP with the help of ATP synthase complex. Complex consists of two major components, F naught and F1. F1 headpiece is a peripheral membrane protein complex and contain the site for the synthesis of ATP and ATP. And F naught is the integral membrane protein. F naught is the protein which is present inside the membrane, so it is known as integral membrane protein, which lead to the, you can say, formation. The passage of proton through the channel is coupled to the catalytic site of the F1 component for the production of ATP. For each ATP produced, two proton passes through F naught from intermembrane space to the matrix down the electrochemical area. Next is respiratory balance sheet. So in this topic we will discuss about the number of ATP produced, the number of NADH, and number of FADH produced, or net gain of ATP we will discuss in this slide here. So we made the calculation based upon the assumption, means in this we will study the glycolysis yield of ATP, then Krebs cycle, then ETS. So NADH will produce 3 ATP, already discussed. So net gain of ATP during the aerobic respiration of one molecule of glucose is 36. Means the answer 36 comes from we add the number of ATP produced by ATP produced by glycolysis, then we add with the Krebs cycle ATP production, number of ATP produced in Krebs cycle, and then at the end electron transport system. So these three component will add on to give that net gain of ATP molecule. Next, fermentation and aerobic respiration. Fermentation accounts for only a partial breakdown of glucose, whereas in aerobic respiration it is completely degraded to CO2 and H2O. In fermentation there is net gain of only 2 molecules of ATP. For each molecule glucose is degraded. So NADH is yield near about 3 ATP, already discussed. This is the amphibolic pathway. What is amphibolic pathway? Amphibolic pathway is that in which the formation and the breakdown reaction will take place simultaneously. So respiration is the amphibolic pathway in which we can see here there is a partial breakdown of substance take place, and through partial breakdown there is partial formation of substances or also take place. So amphibolic pathway has fats, carbohydrate, protein. Protein with the help of enzyme proteases produce amino acids. Carbohydrate will produce simple sugar and glucose. Fat will produce fatty acid and glycerol. So glucose will produce glucose 6 phosphate, and you can see glucose 6 phosphate will convert it into fructose 1,6 phosphate. These are the steps from glycolysis. From this slide we can just conclude a single line that in respiration there is breakdown of substances also take place, but simultaneously formation of some substances also take place during this process. That's why this is known as amphibolic pathway. Respiratory quotient. Now what is respiratory quotient? When a substrate is oxidized in presence of oxygen, then the amount of CO2 evolved by the ratio amount of volume of oxygen consumed is known as respiratory quotient. Means respiratory quotient is equal to volume of CO2 evolved by volume of O2 consumed. So the ratio of volume of CO2 evolved to the volume of CO2 consumed in a respiration is called as respiratory quotient. Respiratory quotient is mainly dependent upon respiratory substrate. Carbohydrate has the respiratory substrate is equal to 1, and fat has the respiratory substrate less than 1, and protein has respiratory substrate is equal to 0.9. So this is the last topic, respiratory quotient.